Oh, hello everyone. This is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and I would be live today. So if you have any questions related to this particular course, you can ask me, and I'll be happy to answer your queries. This session is very short, so I will request you that if possible, I think you should utilize this time because it's only for 1 hour. So uh, please keep posting your questions so that we can start discussing about those. So Ankit Kumar is asking something which is very technical actually. See, currently we still don't have any idea about whether the NPTEL exam is going to be online or offline. I think better we should skip these kind of questions. Most likely it is going to be offline, as like it was conducting the previous year also. But you know that depending on the situation, the pandemic situations, only the IIT Madras is going to take the final decision. And anyway, you will get the full information about the you know about the format of the exams and so on. In the exams, we are going to ask you the 100 multiple choice questions, and it's going to be in 2 sessions. So that much is sure, but whether it will be online or offline, that we cannot comment right now. Um okay, so I think the Rahul has asked very interesting questions. So, irrespective of whether you have the small organisms or the big organisms, the overall process of DNA isolation remain the same. So, for example, in this case, you are saying that you know what will be the mechanism, or what will be the how to isolate the DNA or RNA from a virus, right? So what you are supposed to do is first you have to infect the mammalian cells, right, with a particular virus, if it is a mammalian cell virus or something like that. So then it will going to grow, right? And you know that the virus is going to be present in the cytosol. So first you have to collect the cytosol, and then you have to spin down and remove the protein fractions. And once you have isolated the viruses, then you can actually do the same procedure as we are discussing, as what we have discussed for isolation of DNA or RNA. So it's procedure wise it's same, but except that it might have some additional steps for isolating the DNA or RNA from the viruses. So good afternoon, Ankit, and good afternoon of all other students. So you have very limited time. Please start asking questions so that we will have a very fruitful discussions, and I hope that this discussion session is going to be helpful for you to understand. So Rahul is asking another question where he's saying that genome sequencing is same as preparing the genomic library. No, genome sequencing is different, right? Genome sequencing is where you are actually going to sequence the genome, but the preparation of the genomic library is a part of the genome sequencing. For example, let me share my screens and uh, so that you will be able to understand. Huh? So, for example, you can see that you might have seen, right? When we were discussing about the genomic library preparations, right? What we have done is we have isolated the genomic DNA, and then that genomic DNA is being digested, right, to produce the fragments. So now all these fragments, right, are corresponding to this particular genomic DNA, right? Now, what people are doing is these fragments they are cloning into a suitable vector, and that's how they are getting a genomic library, right? Now, this genomic library is having, for example, this genomic library has 10 to power 5 clones, right? Now, what you can do is you can take the clone 1, okay, and you can sequence the DNA, the cloned DNA, right? Sequence the DNA. So sequence the cloned DNA, right? Now you can take the clone 2 and do the sequencing of the cloned DNA like that. Okay. So if you continue like that, if you sequence all the 10 to power 5 clones, you will be, and if you put them together, right, you will be able to sequence the whole genome. So that is what the purpose of that the genomic library can be a helpful tool for all those who are actually doing the the genome sequencing. So that's why it is easier. If you suppose you start sequencing a genome from the beginning, right? For example, if it is a genome of 10 to power 5 kb, for example, right? Then by the time you reach to 1000, right? By the time you reach to 10 to power 3, the enzyme may not give you the accurate nucleotides. So what you can do is you can just break this into fragments, like 1000 fragments, and prepare a genomic library and then do this way. So that's the way you are going to do the sequencing more accurately. Is it clear? The Prasoon is asking how to make the probe if we studying the chromosome at different stages of the cell division. I think the preparation of the probe is, or the procedure of preparing the probe is same, irrespective of what stage of chromosome or whatever the way you are trying to use that probe. It is not different. Okay. Then we have another question from the Ankit. He's saying that how to isolate protein using the Western blotting technique. So Ankit, we cannot isolate the protein using the Western blotting techniques, because Western blotting technique is a detection technique rather than the isolation techniques. Then I think Soumya is asking, gene resistance to antibiotics are present in the plasmid. How can we make the bacteria less resistant? Is there any way? I think you are mixing the two different questions actually. If you have cloned antibiotic resistance gene in a plasmid, right? Then it is actually going to confer the full resistance uh, rather than the partial resistance. And if you are asking about the reducing the resistance in the naturally occurring bacteria, then that can be done, because if you are actually not allowing the bacteria to propagate for several generations, does not allow them to accumulate the mutations which are actually responsible for sometime conferring the resistance and so on, then probably you can be able to reduce. So probably, if I understood the question properly, I think you are mixing the two different questions. Antibiotic risk, cloning the antibiotic resistant genes in the plasmid and then transforming the plasmid into the bacteria is actually going to give the full resistance, not the partial resistance. Then I think EK Glance is asking why the EcoR1 cuts the DNA between G and A only. See, the EcoR1 is a type 2 restriction enzyme, and it has the S subunit, and that S subunit actually recognizes that particular sequence for cutting. So uh, probably that is the sequence which fits into the active site, and that's how it actually recognizes that sequence, and that's why the EcoR1 only cuts that particular sequence. Then I think Haricharan is asking why we do use CRISPR-9 for sequencing. So I think we have not discussed about the CRISPR technology in this particular course, and it's important to keep your questions limited to this course only. I think there is a full course could be possible on the CRISPR technology. Uh, so please ask your questions in case you have some more questions. As I said, this session is very limited. It's only for 1 hour, and the 1 hour you will get an opportunity uh, to discuss your problems. Um AK Glance. I really don't know what is your name, but I think you are asking. I should explain you the structure of PBR322. Uh, PBR322. I think it's better to understand what you could not understand from the structure of that particular plasmid. Then we can discuss that particular aspect more in detail. So please let me know what you could not understand. Prasoon, what you want to discuss related to homopolymer tailing? Okay. So AK Glance is asking PBR322 and its restriction enzyme. So I think you might have seen this is what? FAT. Okay. Uh, uh, So PBR322 is having the section enzymes or section sites. So you might have seen the PBR322, the plasmid structure. It has the ampicillin resistance gene. It has a tetracycline resistance gene, and it also has the multiple cloning sites. And apart from that, it has the origin of replication. So if you see very carefully, what you will see is that it has all the features what is required for preparing the plasmid, right? So what you will see here is that PBR322 is having a ampicillin resistance gene on one side and the tetracycline resistance gene on other side, and within these genes also you will see that it has the restriction sites, and apart from that it has discrete multiple cloning site also, and apart from that it has the origin of replications, and so that is what it is there. Okay, and then it has so this is the PBR T2, right? uh, and uh, And as far as the registration site is concerned, it has a specific registration site which are there on the MCQs, or it has registration site which can be within the ampicillin resistant genes or the tetracycline resistant genes. So either of these resistant genes can be used for cloning purposes, and accordingly it is actually going to you can select the particular clones. Now, I think Prasoon is asking about how we can be able to prepare the replica plate. So, for how we can be able to prepare the replica plate uh, from the master plate, right? So, there's a clear cut methodology, right? So, what you have is you have a master plate, right? Where you have the clones, right? Where you have the bacterial colonies that these are each clone, these bacterial colonies are corresponding to the colonies, right? So you have, this is the master plate, right? So what you are going to do is first you are going to put some identifiers, like you are going to put three holes into this gel or in this membrane, right? And so that there it is easier for you to align actually. And then what you're going to do is you're going to take a Wattman filter paper. Sometimes people use the other filter paper also. So then you can take the Wattman filter paper, right? And take the filter paper and put the filter paper on top of this, right? So I'm just showing you that with the separate color, right? So so that it will be easy to identify, right? So then what you can do is you take the Wattman filter paper and put onto this, right? So you're going to have, then what you do is you press this Wattman filter paper with a roller or something. So because of that, all these bacteria are actually going to stick to this red color membrane. So ultimately, what you are going to get is you're going to get a membrane where you are going to have the spots for these three, these three screws, right, or these three holes. And then you're also going to have the similar kind of bacterial pellets, right? So you're going to have some bacterial colony which you're going to stick to this membrane, and then you took this membrane and put it onto another plate. So let me just draw this with another color, right? So for example, you take your replica plate, okay, and on this replica plate you take this filter paper and put that, right? So again, it is going to match it with the screws, right, match it with the points. And then when you put it like this, these bacterial colonies what were present onto the original plate will come onto the this plate. Okay, so that will be a replica plate, and you can use this particular membrane also for the screening purposes. So this is going to be your replica plate which you are going to use for all subsequent experiment without disturbing the master plate, and then you can just keep doing the same way, right? These spots what you use, these spots are actually going to helpful in terms of aligning the membranes with the original membrane, and that's how you can be able to do that. Okay, I'm sure it's clear, right, person? uh, and why it is significant to make the replica plate? Because it is significant because you don't want to destroy your master plate, right? You have prepared the master plate with lot of efforts, so that master plate are you should not destroy, because while you are doing all these procedures, you probably could bring the contaminations and you might destroy uh, the the colonies and its morphology as well. So better to work all with the replica plate, and once you got something, then if you want to identify, go back, right? Then you go back and identify the clone based on the orienting the the identifier screws with the onto the master plate. Um, does mechanical sharing of DNA have any advantage over restriction enzyme method for cutting the DNA? Okay, so if you want to use the mechanical sharing, right? If you want to use the mechanical sharing versus restriction enzyme, there are many advantages actually. First of all, mechanical sharing is going to be going to be cheap, right? It does not require the enzyme, right? Number two, the mechanical sharing can be give you the suitable size fragment, right? And on compared to that, the cheap, it is going to be costly, right? If you're using the station enzymes, right, it is going to be costly. The station enzymes could be non-specific, and sometimes it can actually they can be start chewing up the DNA and all that. So because that station enzyme could be could be able to show you the star activity and all that. So that's why it is important that we should use the only disadvantage with the mechanical sharing is that you have to do the lot of standardizations with the mechanical sharing, so that you should generate the suitable size DNA, and that requires a lot of optimization and lot of practice actually. So I hope I have answered most of the questions what you have asked. If you have any more questions, kindly keep posting your questions. Rahul is asking if agarose gel electrophoresis is the only method to separate the fragments. No, it depends on the type of fragment what you are isolating. So you can actually have the different types of technique what you can use. So as far as the DNA fragment, I hope you're asking about the DNA fragments only. So if you are asking about the DNA fragment, right? DNA fragment could be very small or it could be medium size or it could be large. If you have a very small fragments, like if you have a fragment of, like for example, 1 to 10 nucleotides long, right, or single nucleotide even, uh, then you are not going to use the agarose gel electrophoresis. Then what you are going to use? You are going to use high resolution page or polyacrylamide gel electrophoresis. That is, those are also called as sequencing gels. This we have discussed when I think in the later modules. I think we are going to discuss about the sequencing gel. But if it is a DNA of 100 base pair, or if it is a DNA of 1 kb to on onwards, then you can very easily use the agarose gel for separations, and you will get the nice separation of 100 base pair to any other sequence. Okay, so depending on the size, because the agarose has low resolution, it's good for the DNA, but it's not good for the small DNA actually. So for the small DNA, you should use the uh, the high resolution page gels or the sequencing gels. Then Haricharan is asking, what are the easiest cells for culturing? The easiest cells for culturing, it depends what kind of easiness you are asking. If you are asking about the robustness of the cell, like it will not going to, it will be going to be uh, resistance for any kind of handling issues and all that. So I think one of the very robust cell is HeLa, right, which is a cervical cancer cell. So if you use the HeLa, I think it is going to be very, very robust. Even if you don't provide the serum for 24 hours or 48 hours, HeLa will still be able to survive. None of these mammalian cell culture cells are going to withstand any kind of contaminations or something, but even like macrophages also, like if you contaminate them, then they are not going to survive. But uh, as far as the robustness is concerned, right, where you can actually have some kind of mistakes, for example, if you want to learn the cell culture techniques, then you should use the HeLa cells. And that is going to withstand some kind of mistakes from your side. Is it clear, Hari, Sharan? Now, Haricharan is asking how we detect the virus or the mycoplasma, right? So detection of the virus, detection of number one, the virus. So detection of virus is actually is easy, right? Detection of virus you can do by many of the immunological assays. For example, you can do the agglutination assays, right? So you know that the viruses are having interaction with the cells, so they can actually, you can use the agglutination assays or you can use the precipitation assays. So if you do the precipitation assays, you can be able to detect the viruses. Apart from that, you can also do the PCR, right? So if you know the kind of virus is present, then you can do the PCR. Number two, mycoplasma. Mycoplasma also, there are a couple of ways you can do the mycoplasma detection. So you can do the PCR, and the PCR should be able to detect the mycoplasma. Number three, he's asking about do microbes or cells in different media expresses different protein. So if you keep the mammalian cells right in the different media conditions, like different media conditions, uh, I think they will express some proteins because it depends on the conditions. If suppose you are using the rich media, right, then also it is going to give you some stress, right? So rich media is going to give you the stress. For example, rich media means suppose you are using the high glucose. So if it is a high glucose, it is actually going to give you the stress. Similarly, if it is a low media, or if it is a less rich media, right? For example, you don't have the serum, right? If you don't have the serum, for example, if you are using a media which is without serum, then also it is also going to give you the stress. So but the kind of stress what you are going to get in the case of less rich media versus high rich media is going to be different. Here it is going to be a osmotic stress. For example, you have a very high quantity of glucose, so that is going to give you the different types of stress. It's like, a person who is suffering from diabetes, for example. So either if you feed the person well, right, he's going to develop the diabetes. If you feed the person less, he's so diabetes is actually going to cause the stress to the person, right? Ultimately, the diabetes may cause the death of that individual as well. But uh, on contrary, if you are using the media which is less rich, then it is actually going to cause the malnutrition. Like it is going to cause the malnutrition in the individual, and the malnutrition is also going to cause the stress in that particular individual, and the malnutrition also could be could cause the death of that particular person, and the stress in the diabetes is also could cause the death. So if you keep changing the media, right, it is keep changing the expression levels of different proteins. Some of the antigens also could change, and that's why it is important when you do the experiments. It is important that you should do the experiment under the identical media conditions throughout, so that you do not see the media induced expression changes or media induced responses. As far as the product recovery from the insect cells or the mammalian cells is concerned, it is the same, except that the purpose of both the cell lines are different. Insect cell cultures are good for production of the mammalian cells also, and they are very good. So, as far as the product type is concerned, I think both of the things can be able to produce the cells, produce the similar kind of products. Um, so Rahul is asking why the E. coli is used in the most of the biotechnology experiment. Why not other bacteria? See, you use the organisms by the very simple criteria that you know each and every detail about that particular organism. So, as far as the experimental bacteria is concerned, I think we are using the E. coli because we know each and every detail about the E. coli. What kind of diseases it can cause if it if the person got exposed to the E. coli, what could be the different types of resistance are present, what are the different way in which you can be able to deliver the DNA. So all these, since all these technologies are being evolved by doing the experiment onto the E. coli cells, that's why the people are using the E. coli cell. And culturing is also very easy, like E. coli get doubling time is very fast compared to the other bacteria and so on. So that is the only reason that people are using the E. coli for the most of the biotechnology experiments. Uh, I think this course is not for gate preparations. Definitely, it can help you to prepare some content, but as far as the book is concerned, I think if you go through with all the basic books, right, Brown and if you read the basic biotechnology or cloning books from something, I think that should be good enough. Um, Haricharan, I think we cannot produce the therapeutic protein. We do produce some of the therapeutic protein in the prokaryotes, but only with the condition that they should not have the post-translational modification. If those proteins are also required, that it should have the some kind of glycosylation pattern or some kind of that kind of glycosylations or other kind of modifications, then those modifications are not possible in the bacterial cells, and that's why those will not be going to be considered for therapeutic usage. So you can produce the therapeutic proteins in prokaryotes only if such type of post-translational modification is not required. How do we create the artificial plasmids? We have already created artificial plasmid, right? PUC19, PUC18, PBR322, all those are artificial plasmids. If you want to generate or prepare the new artificial plasmids, you might have to do a lot of work, right? You might have to work out many things. Let me explain you with that presentation. So if I want to make a new plasmid, right? For example, if I want to make a new plasmid, right? What I'll have to consider if I want to make a new plasmid, I have to use the multiple cloning site, right? And the moment I will say, okay, I am going to use this set of multiple cloning sites, then I have to make sure that the DNA fragments what I am going to use should not have any site of internal cutters. So you should not have any type of internal cutters, right? See what you're going to have in the plasmid. You are going to have a plasmid, the MCS, right? You are going to have the antibiotic resistance gene. You're going to have the origin of replications, right? These are the minimum three things you require if you want to make a new plasmid, right? And or you might have to have some kind of selection genes, right? So if you have a selection gene, for example, you can have the gene for the blue white screening and all that. So moment, what you're going to do, you're going to start with the MCS. What are the MCS you are going to use, which are going to be unique, right? The moment you are going to use the MCS, then you are going to use the antibiotic gene. All that remaining portion is considered to be under the gene fragment. And this should not have internal cutters, which means any of your enzyme what are present in the MCS should not be there in the cutter. And then you're also going to have the origin of replication, and you're going to have the antibiotic resistance genes, right? So if you can put all these segments together, right? If you put all the cassettes together, you can be able to make the new plasmid. The only tricky portion about a new plasmid is that the compatibility in this context, that you are going to have the MCS and that should not have the internal cutter, which means suppose I have an eco R1 at this site, I should not have eco R1 here. Otherwise, what will happen? You are going to cut the your plasmid with the eco R1 because I want to clone into eco R1, and then what it will going to do is it is going to break the plasmid into two parts. So if you can manage all this, it's possible to prepare your own plasmids. So now very small time left. Okay, so we only have another 5 7 minutes. So if you have any more questions, probably I'll be happy to answer. And you should take this opportunity and try to ask your queries and doubts, whatever you have. Rahul, I think we have not discussed about the CRISPR-Cas9 system in any of my courses. So better you know. I think I have seen one of the MOOCs course where they are talking about the genome editing and all that. So I'm sure they might have discussed about the CRISPR-Cas. So if you are interested, you can enroll yourself into that particular course, and I think that should be very helpful. So I think with this our time is about to finish, and thanks for your time. And I hope these interaction sessions could be very helpful for you. So we have another 1 minute in case you have any question to ask. After that, we are going to stop this live sessions.